Welcome to this unit, which is on mitosis, meiosis, and gametogenesis for Advanced Anatomy and Physiology at Western Technical College. We are going to start out with mitosis. Now, we did talk about this in another unit very briefly. You've probably had this in general anatomy and physiology. So again, we are going to do another review of this because we want to know the differences between mitosis and something new that we're going to get into called a meiosis. And there are some differences, so kind of know what those differences are within those. So mitosis is division of the nucleus in a somatic cell, or what we call a body cell. And so as you can see here with the PowerPoint, a couple bullets down, it says somatic cell. This is our body cell. Basically, a somatic cell or a body cell is any cell in the body other than your sex cells. So for males that would be the sperm, and for females that would be the egg. So all of our other cells that have a nucleus, this is considered to be a somatic cell, and it has 46 chromosomes. Now, with that being said, 46 chromosomes, we do have some other terminology associated with a somatic cell. It can have 46 chromosomes. It can have this abbreviation, if you will, with a 2N, or it can be called a diploid cell. So when we're talking about mitosis, all of those cells that have this 46 chromosomes are considered a somatic cell, and they will go through this process called mitosis, which is, as you can see there, the division of the nucleus. Now, our cells are in this interphase, which is a resting phase of the cell, which the cell spends 90% of its time in this phase until it has to undergo mitosis. Now, not every cell will undergo mitosis. If it doesn't have DNA, it doesn't need to go through mitosis. And yes, there are cells that don't have a nucleus, that don't have DNA. Also, we have some what we consider to be permanent cells, which don't go through mitosis after growth is done. So as an example of this would be like your brain, your spinal cord, your heart. Those cells, obviously, when they're injured, they don't regrow themselves back. They don't go through this process of mitosis, where on the other hand, our skin cells are constantly dividing or going through this mitosis phase because we're losing them all the time. So eventually the cell needs to get out of interphase, and it moves into mitosis. And these are the four phases of mitosis: prophase, metaphase, anaphase, and telophase. And we're going to go through those in just a minute with the pictures. And then at the very end of this whole process, we get cytokinesis, which is the division of the cytoplasm, which essentially makes two new daughter cells, as you can see down here. And so this is a duplication process, and we should see exactly the same information, the same DNA going into these two new cells and what we get out of it. But here's the deal: there is no gene variation, meaning the DNA is not variated at all. And why do I stress this? Because our next subject that we're going to be talking about in just a moment, called meiosis, which is sex cell division, this does have gene variation, and this is, and we'll get into this, but this is why your brothers and sisters don't look alike, because you have such a big gene variation from your mother and father. Whereas mitosis, though, when it does go through this duplication process, you do get identical DNA. At least you should. So real briefly, going through mitosis, again, I don't want to spend a ton of time on this. Here's a picture of interphase. You have this nice nuclear membrane here. The DNA is in the chromatin form. So if you remember back to our last unit, this is the stringy form of DNA. And then you have prophase. Now, there are what we call early and late phases of prophase. I'm not really differentiating between the two. I prefer that you look at this picture right here. And what we see here is the nuclear membrane or envelope has broken down, and the DNA has duplicated itself and is in the form of a chromosome, which is the X-shaped pattern right here. And so you can see they're kind of all in the area where this nucleus would have been before it would have broken down. And then they go to metaphase, where the chromosomes align in the center. Anaphase, they split apart, so we have two identical chromosomes migrating to either side of the cell. And so we see this kind of space between them. And then telophase is the exact opposite of prophase. So we start to see the nuclear membrane starting to come back, and these chromosomes here will start to unravel and become chromatin, the stringy form of the DNA. And we also see this little indentation here, which we call, you can see it up in this picture too, which we call the cleavage furrow, and that's very unique to telophase. And then the last thing that happens here is these two new cells, these two new daughter cells that are being created, splitted will split apart right here, and once they do, that's called cytokinesis, and that ends the whole process. And so this whole process of mitosis takes about 24 hours for a cell to move through this process. So again, didn't want to spend a ton of time on this. I know you've had probably this in the past, but I do need to cover that because our next subject matter is called meiosis, which is the sex cell division and the division of the nucleus that occurs in the gonads, so the ovaries and the testes of both males and females. And with this, we do get variation when the chromosomes do divide, if you will. With this, what's going to happen with meiosis is that we're going to form 4 daughter cells. So look, think about mitosis, it was only 2 daughter cells. With meiosis, it's 4. And also the chromosomes are going to be split in half. So instead of getting 46 chromosomes, we're going to see 23. So we're going to get a lot of genetic variation, variability with this, as we will see. And so this whole process of dividing these chromosomes is known as meiosis. And then we're going to get into how do we form the sperm and the egg, which is the gametes, and this is called gametogenesis. And we'll talk about that process as we're going through here. But when we're looking at sex cells versus the sperm and the egg, they only have half the chromosomes. So they're 23 chromosomes. We sometimes call it a haploid cell versus a diploid cell, because it's half of what you normally would do. Instead of being a 2N, they're just a small italics N. And I know this is capitalized, but it should be a small italics N right here. But you get the idea. So everything's kind of cut in half here. Now with meiosis, we have the same names as we do with mitosis, except for we have what we call a meiosis 1 and a meiosis 2. And so we call that prophase, metaphase, anaphase, telophase times 2. And there are different things that are taking place in each of those. I will stress that the majority of what we're going to talk about is going to be in meiosis 1. Meiosis 2 is fairly straightforward, very similar to division like we saw with mitosis, so there's not a lot going on with meiosis 2, but there's a lot of stuff going on with meiosis 1. And more importantly, what is that lot of stuff that I'm talking about? Genetic variation or gene variation, okay? So that's really what I want to stress. So just to kind of summarize here, what we have here is we have what's going to happen with meiosis. We're going to reduction of the chromosomes for the sex cells down to this 23 chromosomes. We're going to get a lot of genetic variation, and we're going to take a look at that. So we want to compare and contrast mitosis and meiosis. So just look at this briefly, and then we'll kind of get into more of the details. So over here on the left, you see mitosis here. You have prophase, metaphase, and then you have your anaphase and telophase in here, and eventually you get 2 new daughter cells that are identical. They have 46 chromosomes, okay? With meiosis, you have a lot of genetic variability that's going to take place in meiosis 1 right up here. And then when you get down to meiosis 2, it's going to continue to split, 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 very similar to mitosis here splitting. But what you're left with is 4 genetically different daughter cells, which only have half of the chromosomes. So just know that difference there, and this is kind of what we want to concentrate on. So again, we'll really focus on that meiosis 1, and we'll start there right now. Okay, so with meiosis 1 here, here are the phases, and here are some important things that are happening in each phase. We have prophase, and we'll look at this in a picture as well in just a moment. Let me just read this for you. Prophase, the homologous partners seek each other. So homologous means the same DNA strands will seek each other out, and then what happens is they get these tetrads that are formed, and basically they link together, and I'll show you that. And then we have a little crossover that takes place of the genetic material or the DNA, which is basically a genetic swap. We're starting to see a lot of genetic variability right away in the very first phase. With the metaphase, then these tetrads will align, and then anaphase, the sister chromatids, do not separate and then migrate towards each pole, but homologous pairs stay together. So unlike anaphase and mitosis where they split apart, these chromosomes are going to actually stay together in that X format, if you will, and then move across. Now eventually they're going to split, but not until meiosis 2. So and then eventually telophase, we get division, and then we get cytokinesis, and that kind of ends meiosis 1 and then moves into meiosis 2. So let's look at meiosis 1 really quick here, and then we'll get into some more specific pictures, if you will. So what we see here with meiosis 1 in prophase here is we see the homologous pairs that are pairing with one another right here, and then we get this tetrad formation, which is basically they are homologous and they form together this tetrad. They're basically combining together, and we get this what we call crossover right there, where we're swapping some of that genetic material. And it may be a little hard to see with this photo. I thought it was a little larger, but basically if you look down here, you can see this yellow chromosome now has a little spot of purple on it, and this purple chromosome here is a little spot of yellow, and that's that crossover that we see up here, which they've actually swapped genetic information. And so you know, we get this homologous pairs combining to form a tetrad, and a tetrad is those two chromosomes that are kind of linked together, and then we get this crossover, which is a swapping of genetic material. Now with metaphase 1, they align in the center as you see here. In anaphase, they split apart, but notice, unlike mitosis here, the whole chromosome is moving over versus just half of it. And then we get telophase 1 is where we see the nuclear membrane starting to appear again, but we have the chromosomes that are still all together. They have not unraveled, and eventually this splits apart, and we get two cells that have a lot of genetic variability, whereas mitosis, if we saw the split, they would have been identical, right? So we start to see a lot of this genetic variability. So look at this picture here. This is showing us this tetrad formation and crossovers. So here's our two chromosomes. These are homologous chromosomes, so they're identical, if you will, from mom and dad. And then what happens is they form this tetrad formation where they're together, and they do this little crossover thing here. And once they cross over, you can see that we get a little genetic information from this chromosome to swap over, and the same thing over here. And eventually, when they eventually separate, as we will see in mitosis 2, look at, we've got our gamete or a sex cell 1, 2, 3, and 4, and each gamete or sex cell is going to have a completely different DNA strand for that particular chromosome, which makes it variable, right? It makes this genetic variation for real. And so you can see that that's what we see here with that. And again, this is why you look different than your siblings. This is why everybody has a little different look to them. Now with meiosis 2, what we get is equational division, and equational division is where each daughter cell gets equal shares of chromosomes. So we've already had our genetic variability here. We're not going to see that anymore at this point. What we are going to see though is the splitting up of these chromosomes, so that each daughter cell, remember we're going to have 4 daughter cells, will get 23 chromosomes. So it starts with 46 chromosomes in each 2 new daughter cells at the end of meiosis 1, and then that divides into 23 chromosomes in each of the 4 daughter cells. So take a look at this picture right here. This is meiosis 2, where we get this equational division here. So here's where we left off after telophase 1 right here. So then it goes into prophase 2. Here's your chromosomes with that genetic variability there. Metaphase 2, they split, or they align, and then anaphase, they actually split apart. Now see, this is looking a lot more like our mitosis, right? Where it's going through this alignment, but remember, we already did all the dirty work with all that genetic variability earlier on, so there's really not much left to do but to basically you know, divide these all out equally. And so then when we get down to telophase 2 here, each new cell has only 23 chromosomes, and they split apart. And so each one has 23 chromosomes, and more importantly, again, let me stress this, they are genetically different. Okay, so again, just to recap, this goals is to reduce the chromosomes number by half to 23 chromosomes. Introduce that genetic variability with the crossover, the random assortment in meiosis 1, and the fact that no gametes will be alike due to this genetic variation. So which just brings us to gametogenesis, which is the formation of the sperm and the egg, known as spermatogenesis or oogenesis. Now we don't use the word egg here, we use the word oocyte or ovum for that egg, and we'll look at that here in a moment. But this whole process of spermatogenesis and oogenesis does involve meiosis 1 and 2. It also involves the reproduction process, genetic variability, and the end result is a functional ovum or egg and a functional spermatozoon, which is known as the sperm. And both of these have 23 chromosomes in them and are different than any other egg or any other sperm that has been created. So we'll start by looking at spermatogenesis, which takes place in the testes and begins during the puberty years, if you will. And testosterone does play a role in this process of spermatogenesis, along with follicle stimulating hormone as well, which comes from the anterior pituitary gland. And so this all starts, this whole process of generating or creating this sperm called spermatogenesis with what we call spermatogonia cell, which is the stem cell that's going to eventually help create these sperm cells. So remember, stem cells are undifferentiated. They're very neutral. They're not, could go either way, right? And so these spermatogonia cells, these stem cells, are found on what we call the basal laminal wall of the seminiferous tubules. Now, let me show you this. You may have to recap this from general A&P. But here's your testes right here. And inside there is this yards and yards of the seminiferous tubule. And this is where that spermatogenesis is taking place, is in these tubules. So when we look at this stem cell that we're talking about, this spermatogonia cell, where this all starts, is on the very wall of that seminiferous tubule, on the very outside wall. And I'm going to show you a picture of that right here. So here you can see this big round circle here. This is the inside of the, this is the border, I should say. This outside line right here is the border of the seminiferous tubule. This white hole right here, this is the inner space of that tube. And so when we're talking about spermatogonia cells right here, they're always situated out here along this wall right here. Okay, and eventually what we're going to see is them making and going through this process of spermatogenesis to eventually release the sperm into this little space here, so that when a male ejaculates, it can move out. Okay, we'll get into that here in a moment. So what happens here, I'm going to show you a picture of this, but we have basically spermatogonia cells go through division, and they produce a type A and a B cell, if you will. The A cell will stay back at the wall, while the B moves towards the lumen, or the space of the seminiferous tubule. So just like mitosis, it goes through mitosis, if you will, and one of these spermatogonia cells stays back, and the other one moves out. And eventually this one that moves away, we call it cell B, becomes the primary spermatocyte and eventually divides via meiosis to create 4 sperm with only 23 chromosomes. So let's just show you a picture. We like pictures. So way out here is that basal lamina. This is the wall of that seminiferous tubule that I was talking about, or I showed you in a picture earlier here. And here's the spermatogonia cell. So this is that stem cell out here with 46 chromosomes. And so we'll imagine that it goes through division, and type A, remember I said that the type A cell will stay back and the type B cell will go and move on. Now this whole process here, because it's 46 chromosomes, was really this mitosis phase. Okay, so type B then goes down and starts moving its way down, and as it's doing that, it goes through meiosis 1, and it becomes a primary spermatocyte. And as meiosis 1 is complete, we have 2 what we call secondary spermatocytes, which only have 23 chromosomes in them, and that further divides into 4 what we call early spermatids, and these have 4 23 chromosomes, which have a lot of genetic variability with them. So think back to our meiosis that we just went through, and remember this is essentially the sequence that we saw, right? Now what we want to do is look at, how does this sperm it's called spermatogenesis here. So we already went through all the genetic variability in meiosis 1 and the equational division in meiosis 2, so we don't need to go through all that. But what we're getting to now is we have these late spermatids, which start to look a lot like sperm here, and then eventually they become spermatozoa, which are the actual sperm themselves, and these are released into the space of the seminiferous tubule. So basically this slide is kind of rephrasing that, if you will. You know, we have this primary spermatocytes, which divides in meiosis 1 and creates 2 haploid, remember, so 2 cells with 23 chromosomes. And so if we go back, that would be right there. Okay, and then we have a secondary spermatocyte, which moves into meiosis 2 and eventually creates 4 spermatocytes. And again, if we go back, that would be these spermatids right here. And they have a lot of genetic variability. All of them are different. And the spermatids are these small round cells that compact their DNA very close together and protecting it. And these are the spermatids here. So we have these early and late spermatids, if you will. And so you see they have some of this cytoplasm that's still kind of hanging on there. And we'll talk a little bit about that here in just a moment. Let me jump up to this slide. This is the same thing that you saw right here, but with just a little bit more detail, if you will. So let me just show you this again. This is, again, it's the same thing, just with a couple little different details. So here's your spermatogonia cell. This is your stem cell. This divides. There's cell A, we'll call. There's cell B. It goes through meiosis 1, and we get 2 secondary spermatocytes, which have 23 chromosomes. That goes through meiosis 2, and then we get the 4 early spermatids, which have 23 chromosomes, all genetically different. And then we get this late spermatid right here, and then eventually we get our more functional, mature spermatozoa, which are known as a sperm, and they get released, and they're ready to be ejaculated when the time is right. So you can see this process here a little differently. Now I'm going to talk about this, all this yellow stuff right here. That's actually a cell. I know it's kind of weird. All this yellow stuff right here is a cell. There's the nucleus of the cell. We're going to talk about that in just a little bit. So this kind of gives you an idea what we're looking at. So spermatids have 23 chromosomes. They're known as a haploid cell. In this process of shedding this cytoplasma that you saw, that extra cytoplasma that you saw, is called spermiogenesis. And so if we go back to this, right here, I like this one better right here, you can see the cytoplasm that's still left on that. So we need to get rid of that and have these little spermatids mature, and that process is called spermiogenesis. So the spermatids, after they go through spermiogenesis, will eventually become spermatozoan, which are the sperm, and the head of the sperm have the genetic information. The mid piece is the metabolic, which has the mitochondria, and then the tail is the locomotion part, which helps make it swim, if you will. So if we look at this sperm, this is the one that we want to look at, and you can see this process of maturing through spermiogenesis here, going from this immature spermatid to this functional sperm. So here's the head. Inside there has the genetic information, the 23 chromosomes. There's a little acrosome. This little black head on the sperm right here is called an acrosome, which has the digestive enzymes to penetrate the egg, if you will. We'll probably be talking about that more at a different time. I think we talk more about that in reproductive physiology. But and then here's your mid piece, which is going to house the mitochondria, which produces a lot of ATP. And then finally you have your tail, which is the locomotion part. So the head is the most important part for fertilization with the egg, right? It mixes with the egg's DNA and you start to create a new cell. The mid piece is really important because it's producing a lot of energy for the tail to propel itself. Okay. So, I can't say this. Sustencular cells. That's a hard one for me to say. Sorry. are cells that we were just covering just a moment ago. So if we go back here, you can see these cells. These are these big yellow cells right here, and this is the nucleus here. So they play a pretty important role in really kind of keeping everything together here and making sure it's real tight, if you will. What they do is they form a what we call a blood-testis barrier. We don't really want the sperm to be introduced into our bloodstream. That could cause an immune reaction. So these cells here will help with that process of making sure that it stays really tight. Those sperm don't go away. The sperm go where they need to go, which is in the lumen, or the opening of the seminiferous tubules. They also help create what we call testicular fluid, which helps to give nutrients to the spermatids and create some pressure differences within the testes as well. And so again, you can see these cells right here, big yellow cells here with the little nucleus as well. So that really summarizes spermatogenesis. What about the female and the oocytes? And so the oocyte is the sex cell of the female when it is not fertilized. It's an immature cell, where the ovum is a sex cell, but it is considered an ovum when it's been fertilized, and it's what we consider to be a mature cell. So females, you are born with about 7 million, all the oocytes or eggs that you need to ovulate throughout your lifetime, and 2 million of them escape program death, and they remain there until puberty. So you've created 7 million, only 2 million are left, and at the time that puberty begins, then at that point you have about 250,000 left. So you start with a lot. They kind of arrest, if you will. They stop growing until you hit puberty, and then they start going through this maturation phase again. And only one egg is recruited every month to be ovulated, and this is because of a surge in LH, which is luteinizing hormone, which comes from the anterior pituitary gland. And ovulation typically occurs 1 to 3 years after your very first menstrual cycle. So it's not necessarily that you had your very first menstrual cycle in middle school, we'll say, and you started ovulating. It takes just a little bit of time for that to kind of really rev up and get going with ovulation. And so you obviously are not going to use 250,000 oocytes throughout your lifetime. You figure your span of ovulation really is only about 30 to 40 years, and it's getting younger as we age, and with different things that are happening in our society and how we're changing. So you by no means are going to be using all 250,000 of these. So this process of developing an egg or an oocyte is called oogenesis. So very similar for the most part. It takes place in the ovaries, and it takes place with a beginning with an oogonian cell, which is a diploid cell, which has 46 chromosomes. And again, this takes place in the fetal period, so this was taking place before you were even born. And what we have happening is we have these primordial follicles that begin to develop, and then eventually a primary oocyte is created as well. And we'll look at a picture of this in just a moment. The primary oocytes will begin the first mitotic division, and they will eventually stall in prophase 1, and it does not complete until puberty. So you were born, this is all fetal development here. You were born with these primordial follicles. A follicle is just cells around the egg or the oocyte, and around these follicles are around the primary oocyte. And the primary oocyte you've created, it's like an immature egg, and it stalls in prophase 1 of meiosis, and then it doesn't go through the rest of meiosis until puberty, until you're ready to ovulate and whatnot. And this is the reason for that is just it's all hormone related. Okay, it's all hormone related. That's why this is happening. So if we look here, we got these mitotic events of a primary oocyte, if you will. And what we have here at birth, you have this oogonian cell, stem cell, so very similar to the male. You have 46 chromosomes. And then it arrests, okay, and it stops here. and infancy and childhood, it stops here, right? So you have this primary oocyte that's just kind of hanging out until you hit puberty, okay? And once you hit puberty, then once a month, one egg is chosen to move on to go through meiosis 1 and meiosis 2, okay? And here we get down here, and we'll get into this a little bit more, but here's our 1, 2, 3, 4 different genetically different cells, so very similar to what we saw with the sperm, okay? But only one of them, typically, only one of them is going to go on to be a functional oocyte. Now, what are follicles? Follicles are cells around the oocytes that help mature them. So we'll see this as we progress here. The follicles get bigger, they get more mature, and that's going to help with developing a functional oocyte as well. But right now, where we're at is right here. You're just right there, your infancy and childhood. You've made all 250,000 of these primary oocytes. They have 46 chromosomes. They're just waiting for you to go through puberty. Okay. So, meiosis 1 is completed. You eventually get 2 haploid cells, which are your first polar body cells, if you will. And you get a secondary polar body, which is a little bigger. So let me just go back to this picture here. And here's where we're at. So meiosis 1 is complete. We have all that genetic variability. Here's your first polar body. Now this, compared to this, is much different. Why? This doesn't get all of the stuff. When it goes through division, this one is immature. It's weak. It's not going to be able to be fertilized. But this one here will. So these two have 23. So this is what we call our first polar body, has 23 chromosomes. This is our secondary oocyte, which has 23 chromosomes. And this is the one that's going to go on to mature and become viable, meaning that if it was fertilized, it could do the job, okay? All right, let me go back up here. The first polar body continues to meiosis 2 and develops 2 more first polar bodies. So let's go back here. So there's our first polar body. It goes through meiosis 2 and develops 2 genetically different polar bodies. But again, very immature, very weak, not going to be able to sustain fertilization if it occurred. So really they're just kind of a wash, to be quite honest with you. The secondary oocyte stalls in metaphase 2 and is ovulated. Very important right here, okay? So it stalls in meiosis 2 right here. There's our secondary. It goes down, it stalls. Now, it is ovulated at that point. If it is not fertilized, it stays like this. Okay? Meaning if the egg doesn't fertilize it, it stays like this, and you menstruate it out, and it's washed away. Okay? But if a sperm fertilizes it, then it completes meiosis 2. This is very important. It completes meiosis 2. And if that happens, we get this secondary polar body, which again is useless, and we get an ovum right there. That's that mature egg that has been fertilized. Okay. So you can see that there is some pretty big differences between, spermatogenesis and oogenesis that you do need to know. So I think it's really important to start with meiosis. What is that? And then apply it to spermatogenesis and oogenesis and know the differences between them. Now we're not quite done here. So the sperm penetration, if no, the egg will deteriorate. It will be you'll menstruate it out. If yes, meiosis 2 is completed, and that secondary polar body forms like we saw with the picture. Sperm penetration here, we get this large ovum, we get that smaller secondary polar body, which does nothing for us, and so the end result of oogenesis is 3 tiny polar bodies, non-functional, and 1 large ovum, which is functional, that can be fertilized. Now when this sperm and this egg meet, and they fertilize and they create the very first cell of your body, that is known as a zygote. Now we will talk more about that in another lecture, but I just wanted to mention that. Okay, so same picture as we saw before, but now let's look at follicles. Okay, follicles are the cells around the oocyte, and they do play an important role. Follicular phase, if you will, is the period of the follicle growth. It occurs 1 to 14th day of the menstrual cycle, so it begins right away after you have your menstruation in the very first part of the menstrual cycle, and then it's done after about the 14th day. Okay, it's gotten to a mature phase, so we'll look at this in just a second here. Follicle cells have one layer, where a secondary follicle has 2 or more layers. So the more layers they have, the better. It's going to help protect and nourish the oocyte. And we have different stages of maturation for different structures. So let me just show you this picture. This is the ovary here. Here are the primary follicles. So you can see the little oocytes here, and they're really small, but they have this little teeny little follicle around them. And one of those cells is picked to mature, and it goes through the process of oogenesis. And look how the follicle gets a little bigger. Okay, it's a little thicker, and then it gets a little thicker and a little thicker, and eventually we get this big follicle here. We call this the graphene of the mature follicle. It has this big blister-like look to it, and this is a very mature follicle that's really nourishing that's what we call a secondary oocyte at that point, until it's ready to be ovulated, as we see right here. There's a lot more to this story than what I just said, because this follicle here does not ovulate. The only thing that ovulates is the egg. You can see that. So this big follicle right here kind of sticks around and eventually becomes what we call the corpus luteum, which we're going to talk about more in a bit here. So here are all the different follicle phases. Okay, we have primordial, which is 1 layer. Okay, so if we go back to this picture, you can see these primordial follicles with 1 layer here. We have a primary with 1 layer. If we go back, go back here, these are primary, so they get a little bigger. You can see the cells get a little bigger. These are our primary follicles right here. We have our secondary late, which has 2 or more layers, so that would be, something around right here, these secondary late follicles. And then we have our graphene or mature follicle, and this is our graphene or mature follicle, and has, as you can see right here, it has this antrum, which is a fluid cavity that appears, and we have this corona radiata. So if I go back here, this is that corona radiata, which is these cells right here that eventually are ovulated with the secondary oocyte here. But this is that graphene or mature follicle. And then finally, as we saw, the corpus luteum, which is going to be what's left of this graphene or mature follicle right here. And that will stick inside of the ovary for a certain period of time, which I'm not going to get into because that gets into our other unit called reproductive physiology. So don't want to go into that just quite yet, but you get the idea. And our last picture is really just showing you that follicle process. Here's your primordial follicles, then it gets a little thicker, it's primary follicle, then secondary, and then you have this graphene or mature follicle here. You get that ovulation, and what's left of that mature follicle is this corpus luteum. Okay, so that is this in a nutshell. So I think what's really important is to really go back, understand meiosis. What's the point of that? Remember, what is the goal of meiosis, and apply it to both the male and the female producing the sperm and the egg. It's to get 4 genetically different cells at the very end. But as we know with the female, they only get 1 that's useful, as we saw, right? So there's a lot of differences. So really take a piece of paper. What I would do is really take a piece of paper out and really compare and contrast the differences between these and really understand the most important talking points as well. If you have any questions, let me know. But we're going to follow up with this because we have like reproductive physiology, we have genetics we have to go over. So this stuff is really important as like as a stepping stone to our next level. So if you don't understand like the whole genetic variability and what's the purpose of that, it may make genetics and reproductive physiology just a little bit trickier for you. So really study it up. Let me know if you have any questions.